Thank you very much, Kathy, for the introduction. And I'm really honored to be here with all of you. And I'll just give you some update about the work that we've done since, securing the grant. Can you see my screen?
Yes.
All right. So, yeah, I'm a basic biologist, so I'm mainly interested in the basic science of, let's say, proteins in particular. My lab is mainly cancer biology, but I was lucky to introduce to meet Kathy, and she actually encouraged me to apply for this grant, and we're quite lucky to get this grant through the foundation. So the work that we submitted for the grant was actually to do some structural and functional investigation of the ZC4H2 protein. And I've just got a video, a few seconds here. What we're trying to do is understand life at the molecular level. So, like the speakers earlier spoke about the genome or DNA, and these produce the proteins, which are the functional units, in a cell. And these, will have impact on the, organelles, the organism in total. So what we're trying to do is understand life at the molecular level. And they have, made my life, easier by introducing the basic biology of this. So we know that the cells has DNA, and like Julian said, this is got transcribed to messenger RNA, this intermediate here between, and that the RNA get translated to protein, which is the functioning unit of a cell. So the protein within a cell has a certain function. And if we have a pathogenic, protein, such as the case here with the mutation with the ZC4H2, that may impact, certain diseases. In this case, we're saying about the ZOR diseases. So again, our main interest is to study the structure and the function of the ZC4H2 protein. And why does it matter? Because, if we understand, what's the, how does the protein function, potentially that can help to, producing therapies to treat this disease. So as Julian implied earlier, the ZC4H2 is, a protein of a function is what we call a transcription factor. What does that mean? Its role is actually to bind to the DNA, as you can see in this figure here. And, and that is to induce the expression of other genes. So it binds to, the genome, and then it transcribes other gene to messenger RNA, and those get translated to protein. You can see initially the amino acid chain of polypeptide, then that get folded into a three-dimensional structure protein, which has a function. Okay, so the impact of ZC4H2 when it's mutated, it might impact all the genes that it meant to be translating in a certain cell type, which is, neuronal cells. So the main objective, this is here just illustration of the protein. The way we illustrated here, it has different domains, and each of those domains within the protein has a certain function. For example, here where it says zinc finger, it binds zinc to allow it to bind to the DNA to perform this function, where it allows it to bind and then induce expression of other genes, other targets. And as Julian again showed you earlier, we have cells early in development, which are stem cells. So these are undifferentiated cells which have been committed to a certain lineage or fate. And during development, they go into certain lineage and, commit to a certain fate. In this case, for example, in the neuroectodermal cells, they become neuroendodermal progenitors, then they become neuronal progenitors, and they differentiate to what we call post-mitotic neurons, cells that do not, divide any further. They become neuronal cells. So it's really important, to understand, the role of the ZC4H2 early in development. And there are some studies that shown its main function is early here during progenitor cells, perhaps. And that may impact, if you have an impaired protein, that may impact how the neurons are developed. So one of the main objectives of the study that we submitted, to do is to do this kind of work. So again, Julian have shown that, we've got also those cells through the Korea Institute. These are what we call induced pluripotent stem cells. So these have been taken from either skin or blood of a patient. And what we do, or what the Korea Institute have done, they could revert back. These are differentiated cells. They became blood cells or skin cells. And, this technology came, available around 15 years ago, which you could take a differentiated cells and revert it back to a stem cell stage. And the advantage of that is for disease modeling, then you could take those stem cells and can differentiate them to any type of cells. In this case, we're interested to make neurons because these are what's impacted in the disease. So we could grow them in petri dishes in the lab, and we could make them to those intermediate, which are neuroprogenitor cells, excuse me, and then we could make them to terminal differentiated neurons. And again, what we want to do is take those induced pluripotent stem cells from a patient with the disease compared to those with healthy controls. And the healthy controls is just iPS cell line from somebody with a matched age. Okay. And we're going to study what we call the transcriptome and the proteome across these different developmental stages. How does all the messenger RNA, this is literally every gene within that's encoded by the genome that's got transcribed into RNA. How does that change when you have the mutation of ZC4H2 versus those healthy control? We look at that using a technology called RNA-seq to sequence the transcriptome and mass spectrometry to look at the proteome, and we compare to say those with the disease, how does that vary compared to the healthy controls. And we do that at the different developmental stages, when there's still a stem cell stage, and when they're neuroprogenitor cells, and when they become fully differentiated. And we think that's important because, as I said, there's literature that suggests its important role is here, early in development, perhaps when there's still stem cells or progenitor cells, and the expression, the amount of the ZC4H2 protein actually drops as cells become neurons. That means it may have less of a function when the cells are already differentiated. So if there is an impairment, it most likely to be at this stage, which we want to capture it. So this is one of the three objectives that we aim to do in the lab. The other one is that these transcription factors usually do not function independently. They have partners. When they bind to the DNA, they recruit other proteins for it to be able to perform a function. So what we're going to be doing some specific mass spectrometry analysis to determine what are the factors, what are the cofactors and the partners that the ZC4H2 need to bind to do its function, and how does that vary in healthy versus controlled disease, again, to understand function better. And this is here again, and the third part is to understand the structure of the protein. So in here, if you can see here a schematic of the protein, this is for what we, a region which is cold, we refer to it because of the structure. This is where it binds zinc to allow it to bind to the DNA. And this is just a platform here that compares this, protein across different species. And in blue, it shows you that this region is highly conserved. So you can see here, this region is highly conserved across species, which imply it has important function of the protein. So if you were to have a mutation, perhaps in this region, which will impair the way it binds to the DNA, it will be more impactful, perhaps if the mutation was in this region, which is less conserved. This is an artificial platform that can predict how the protein fold and form a three-dimensional structure based on sequence. Of course, we're doing experimentally this and trying to, to see the structure of the protein. But as I said, this is what it predicted, that this region is cold, formed this kind of structure, and actually it dimerizes with itself and forms the homodimers. This is the region here where it binds to zinc. And as I said, it's important for it to perform its function in DNA binding. So in here, we're doing some zinc binding assays to, because this has not been shown before that although it's ZC4H2 family of protein, but no one studied particularly the ZC4H2. So we'll be the first to show it actually binds zinc. And we're doing some crystallography study to determine the structure of this protein. So, as I said earlier, this family of protein is being studied, but not the ZC4H2, and the way it binds zinc has not been shown before. So this is the work we're doing in the lab. And the way we do it is actually we clone the part of the genome or the gene that encode that, the ZC4H2, and we put it in what we call a plasmid. We introduced this plasmid into a bacterium because it, replicates quite a lot, and we can produce this in mass amount. We lost the bacterium, and we use some biochemical assays to purify our protein of interest. In this case, is the ZC4H2, and we send this for structure analysis to understand how the structure of the protein, how does that change when there is various mutation, how does that impact the structure of the protein, and that we can help us understand the function if it's mutated. This is some data we generated in the lab. The way we purify this protein is, as I showed you earlier, but this band here indicate that we get a purified protein. This is the full length, the whole protein here. And we're also interested to do some structure analysis of the different domains that called core region here and zinc. This is the called core region here. Again, we purify successfully. And now we've sent those to our collaborator at Harvard Medical School, where they're doing some crystallography and NMR studies to determine the structure of the full-length protein in those domains. We've done some work, as I said earlier, to show it's actually binds zinc. So this assay here, we take our purified protein, we could inject zinc, and the machine just measures the temperature if there is any interaction between the protein and zinc. And this here shows you, those peaks shows here that there's actual interaction between the protein and zinc, verifying, for the first time for ZC4H2 that actually, bind zinc. So, sorry, this is clicked here. Yeah. So in summary, what we believe that, our structural studies will shed insight into the protein function, and perhaps, see how those various mutation impairs its function, particularly early in development. So by, having a more clear, like data on how the protein fold and the structure of the protein, perhaps we can understand better how mutation impairs its function in early development. The work we're doing, where we're studying the transcriptome and the proteome across the patient-derived, neuroprogenitor cells and neurons versus those of the control, again, will help us shed insight into what are the pathways and what are the other genes that are regulated by the ZC4H2 that may be impacted by when we lose the function of this protein and at the different developmental stages. And why is that important? Because, again, a better understanding of the basic biology, the protein structure, the protein function can potentially help us develop therapies for this disease in the future. So I just finally want to thank, the people who contributed to this work, Dr. Rania Najim, the postdoc in my lab, and Dr. Said's lab, where she actually did all this work in purifying the protein and the zinc measuring data. Our collaborator at Harvard Medical School, which is doing all the structural analysis for us. And of course, for Catherine, for encouraging me to apply for this grant and get involved. It's, an honor to be able to contribute, even if, understanding the basic biology of the protein. I want to thank the Foundation and the Orphan Disease Center and the Korea Institute, the Foundation for funding the study, the Orphan Disease Center for managing the grant application, the Korea Institute for generating the cell line and providing the cell line. And of course, Dubai Health and Al Jaleel Foundation, where I work, and they also, fund some of the other work I do. I'm happy to take any question if there is any.